Assalamualaikum and salam sejahtera. The second learning unit would be on protein. Okay, these are the learning objectives that everyone must be able to accomplish by the end of this learning unit. The first one is to define and classify amino acids, followed by memorizing the chemical structure of the amino acids. Next is explaining peptide bonds. What are peptide bonds? And then give example of these peptide bonds. The fourth one is defining and explain gelation. Next is explaining chemical alteration of amino acid and what are the changes due to the chemical alteration of amino acids. The sixth learning objectives is to define and classify protein. So how many classes of protein are there? And then next one is explain protein hydrolysis and denaturation, and what are the factors that leads to protein denaturation. Next is identifying factors which affect protein solubility, and finally defining and classify types of emulsions. So what is amino acids? Okay, the definition of amino acid: they are the building units of protein. What is protein? Okay, proteins are polymer of these amino acids linked together by peptide bond. Okay, so there are about 300 amino acids occur naturally. Okay, but only 20 of these 300 occur in protein. So therefore our focus is only these 20 amino acids. So let us look at the chemical structure of amino acids. So each amino acid will have 4 groups which attach to the alpha carbon. Okay, which means carbon next to the carboxyl, COOH. So what are the 4 groups? Okay, the first one is hydrogen atom. Okay, carboxyl group, the second one. Okay, amino group, the third one, and then finally the side chain R. So this side chain R will differentiate one amino acid to another. The rest, this 1, 2, 3 groups are the same. So since the side chain R are the deciding factor for intra and intermolecular interaction in protein, so the amino acids can be classified as either polar or non-polar amino acid, and based on the chemical classification, it is either neutral, basic or acidic. And if we look at in terms of their nutritional or physiological roles, amino acids are classified into either essential, semi-essential or non-essential amino acids. Okay, so at physiological pH, which is around 7.4, the carboxylic group is dissociated and it will form negatively charged carboxylic ion, COO minus. And then the amino group, okay, the amino group is protonated and it will form positively charged ion NH3+. And this will form ion having both positive and negative. We call them Zwitter ion. So neutral amino acid, okay, which is monobasic and also monocarboxylic, will exist in solution as Zwitter ion. Okay, this is in aqueous solution. Okay, so it contain both positive and negative charge. Okay, first we will look at classification of amino acid based on the chemical structure. Okay, so it's either neutral, basic, or acidic. Okay. So let us first look at neutral. Okay, neutral amino acid, and the simplest form of amino acid would be glycine, where the side chain R is hydrogen. The second example of amino acid which are neutral amino acid is alanine. Okay, alanine, the side chain R is methyl group. Okay, CH3. And the third neutral amino acid is branch. Okay, branch amino acid, where the side chain R, okay, if it's isopropyl group, then it's valine. If it's isobutyl group, then it's leucine. If it's isobutyl group, then it's isoleucine. Next is neutral amino acid containing sulfur, and there are only two of them, which is cysteine and methionine. Okay, so we will look at this structure in the later slides. Okay, the fifth neutral amino acid containing hydroxyl group is serine and threonine. So the next neutral amino acid, which is aromatic, is phenylalanine. Okay. Second one is tyrosine. The third one is tryptophan. Okay. Next neutral amino acid is heterocyclic. Okay. Example is tryptophan, which contain indole ring. So the only amino acid containing indole ring. Second one is proline. Okay, this slide shows the mentioned amino acid just now. Okay, so this is the only amino acid with indole group, which is tryptophan. Amino acid containing sulfur, okay, methionine, as well as cysteine. The one containing aromatic ring, okay, phenylalanine. The one is branch, okay, valine, leucine, isoleucine. Okay, so these, in order for you to memorize, I will upload the video in elip in order for you to easily memorize these structures. Next, we move on to the second classification of amino acids based on the chemical structure, which is basic amino acids. So we have covered the neutral amino acids. Next, we move on to basic amino acid. So basic amino acid will have two or more NH2 group or nitrogen atom, and this nitrogen will act as base. Okay, the one that can bind proton. So at physiological pH, basic amino acid will be positively charged. Okay, so they will be positively charged. So example of basic amino acid are lysine, arginine, histidine. Okay, so they are charged. Okay, charged amino acid. Okay, so this is basic amino acid. Okay, positively charged. Okay, histidine. Okay, arginine and lysine. And the acidic amino acid will be negatively charged. Okay, example is aspartic acid and glutamic acid, only two. So aspargine and glutamine, although it sounds similar to the negatively charged acidic amino acid, which is aspartic acid and glutamic acid, in fact actually the amide form of aspartate and also glutamate, in which their side chain, okay, the COOH group becomes amidated. Okay, so therefore they are neutral amino acid. Okay, asparagine, glutamine. Note that the name, okay, this one, okay, ASPGLUNYS is the abbreviation of amino acid that you should know. Okay. Okay, next we move on to the second type of classification of amino acids based on their intermolecular or intramolecular interaction. Okay, so it's either polar or non-polar. So polar amino acid, okay, in which the R side chain is hydrophilic group. Okay, so they can form hydrogen bond with water. Okay, so this hydrophilic group can be OH, for example in serine, threonine, and also tyrosine. It can be the one containing sulfur, okay, for example cysteine, or the one that can contain amide group like glutamine and asparagine, and also NH2 group or nitrogen. Okay, the basic amino acid. Okay, for example lysine, arginine, and histidine. And another side chain that can leads to polar amino acids are the carboxylic group. Okay, so acidic amino acid. For example, the aspartic and glutamic acids. Next, we move on to the non-polar amino acid. Okay, so based on the non-polar name, of course, the side chain R is the hydrophobic group. Okay, means cannot form hydrogen bond. So there are 9 amino acids that are non-polar. So glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline, and methionine. So you have to memorize this. So there are only 9 non-polar amino acids. Okay, the rest are polar. So easy for you to remember non-polar, and the rest are polar. So these are the structure group based on the polar and also non-polar group. Okay, next we move on to the third classification of amino acid based on their nutritional properties. Okay, so it's either essential, semi-essential, or non-essential. Now, protein are required as building blocks for protein biosynthesis, and it also contribute as flavor of food. So therefore, they are classified based on these 3 groups here. Essential amino acid means the one needed for your body for the biosynthesis now. Okay, semi means you need it partly. Okay, because part of it can be synthesized by your body. Okay, and then of course the third group is non-essential, means you don't need it, okay, from diet, because your body can synthesize. So how to memorize this? Okay, very easy. This is the acronym that you can help you to memorize. Villa, HM, 10,000 pounds. V, valine. I, isoleucine. L, lysine. L, leucine. E, arginine. H, histidine. M, methionine. T, tryptophan. TH, threonine. And P, phenylalanine. So only A and H here, okay, arginine and histidine, is semi-essential. Okay, formed in the body but not sufficient. Okay, especially in children. Okay, so the rest are all essential, and the one that are not mentioned in this Villa HM 10,000 pounds are non-essential. Okay. So these are the non-essential amino acids. Okay, they are synthesized in the body. Okay, so you don't need it from your diet or from food. So these 10 amino acids are glycine, alanine, proline, serine, cysteine, tyrosine, asparagine, glutamine, aspartic acid, and glutamic acid. So this table here shows the amino acid composition in different type of food source. Okay, so we have fresh peas with flour, chicken breast, beef steak, whole egg, cow's milk, human milk, okay, cod fillet. Okay, so the most important source, if you notice here, of protein are grain, okay, oil seeds, legumes, then it followed by meat, okay, meat and milk. Okay, so you see here, the value is quite high. Okay, especially we focus on the essential amino acid. Okay, Villa HM 10,000 pound. Okay, next we will continue with peptides and protein. So we have covered there are 20 amino acids that are commonly found in protein, and these 20 amino acids are linked together through peptide bond, okay, to form peptides and protein. And if the number of amino acids join together via peptide bond are less than 50 amino acids, we call them peptides. Okay, if the amino acid is more than 50, we call them protein. So how does the mechanism of this peptide bond formation? Okay, so if you remember, the basic structure of amino acid, okay, so this is the side chain R, so this is the amine group, okay, this is the carboxyl N, and you have hydrogen here. Okay, so this alpha carboxyl group of one amino acid, so this is one amino acid with another side chain, okay, form a covalent peptide, okay, with another alpha amino group of another amino acid. okay, having R2. So let's say this is R1 and you have another one here, this is R2. Okay, so these two are joined together, and when these two are joined together, one molecule of water is removed. So we'll end up forming dipeptide. Okay, so dipeptide means two amino acids linked by one peptide bond. Okay, so this dipeptide can form another second peptide bond with a third amino acid. So we have another third amino acid here, R3. Okay, R3. So this will give you tripeptide, and this will be repeated, okay, to give you either peptide if the amino acids joined together are less than 50, and if it's more than 50, we call them protein. So this is example of formation of peptide bonds. So we have serine here, one amino acid, and tyrosine here, okay, one amino acid, okay, and they are joined by peptide bond, and then during this formation of peptide bond, one molecule of water is removed. Okay, and then this dipeptide, okay, can form another peptide bond with a third amino acid. So in this case here, cysteine. So it will remove one molecule of water. Okay, so the carboxyl N made up with the amine N. Okay, amino N. So the amino N, we call it as N-terminus. The carboxyl N, we call them as C-terminus. And these will create the backbone of the peptide or protein. And this is where the side chains are. So next we will look at example of peptide. Okay, so example of dipeptide means two amino acids joined by one peptide bond. So example is aspartame. So aspartame is a synthetic sugar. Okay, so it is used in replacement of cane sugar because it is 0 calorie. So nowadays many food are using sweetening agent which are synthetic like aspartame. Okay, so that it gives you no calorie not being absorbed by your body. So this is very suitable actually for diabetic patient. Okay, so how is this composed of? Aspartame is actually from aspartic acid and phenylalanine amino acid. And one thing about synthetic sugar, they are very much sweeter than sugar. Okay, like aspartame here is 200 times sweeter than sugar. So means food manufacturer would only need to use less, okay, to produce you beverages at the expected sweetness level rather than using sugar, okay, sucrose. So the second example is tripeptide. Okay, tripeptide means you have three amino acids linked by two peptide bond. So example is glutathione. Okay, so it's made of glutamic acid, cysteine, and glycine. Okay, so this is commonly found in animal, in plant, in microorganism. Okay, so the function of this glutathione is to protect cells from oxidizing agent. Okay, so this is good antioxidant. Next is polypeptide. So polypeptide means you have 10 to 50 amino acids joined by peptide bond. Okay, example of polypeptides are hormone, okay, insulin hormone. So this is glutathione. Okay, next we will move on to the structure of protein. So protein will only form if there are more than 50 amino acids joined by peptide bond. Okay, so in terms of structure, there are 4 levels. Okay, the first one is primary structure. Okay, so primary structure is just a straight structure. Okay, straight one. Okay, you will have one carboxyl N, okay, and one amino N. Okay, C terminus and terminus. Okay, just straight. Okay, straight chain. Example are lysozyme enzyme. So this enzyme is example of primary structure of protein. So primary structure of protein normally stabilized by intramolecular hydrogen bond. Why intra? Because you only have one. Okay, one unit. Okay, so intra between the side chain of one to another. Okay, one side chain of amino acid to another side chain of amino acid, like this one, very close. So you have the intramolecular hydrogen bond. So the second level structure is secondary. Okay, so secondary structure is further divided into two. The first one is alpha helix. Okay, it's a spiral structure. Okay, this one spiraling. Okay, the one that you see in the primary structure is just straight. Okay, straight one, but it can be curvy. Okay, straight and curvy, but not spiraling. Alpha helix spiraling. Okay, second is beta sheets. Okay, so stack into one another like this here. Okay, so this alpha helix is stabilized by intramolecular hydrogen bond, okay, between one to another, the one that is close enough, like this one. Okay, beta sheets, okay, they are formed from two or more polypeptide. Okay, you see here, one polypeptide, second polypeptide, third polypeptide, and it can be more. Okay, and it will form hydrogen bond between one polypeptide to another. Yeah, okay, the side chain. Okay, so hydrogen bond in alpha helix. Okay, one amino acid. Okay, from hydrogen bond with the fourth. Okay, fourth. This one to the fourth. Okay, so this is 1, 2. Okay, 1, 2, 3, 4. Okay, here. So the stabilizing force for secondary beta sheet are intermolecular, because you have one polypeptide stacked on to one another. So you have so intermolecular hydrogen bond, okay, van der Waals forces, hydrophobic interaction between the non-polar side chain. Okay, so these are the stabilizing force for beta sheet structure. Next, we move on to the tertiary structure. So these are the stabilizing force for tertiary structure. Okay, again we have hydrogen bond, okay, among the polar side chain. We have ionic bond between the charge R group. Remember, there are charge group whether it's basic or it's acidic. Okay, so we have here, okay, ionic bond. We have hydrophobic interaction. Okay, this would be among the non-polar side chain here. For example, here non-polar. Let's say this is one amino acid which is non-polar. This is another non-polar. So the side chain, okay, here will form hydrophobic interaction. Okay, and also van der Waals. And this is example of hydrogen bond. Okay, side chain which are polar. So if you look at this here, okay, tertiary is composed of the spiraling alpha helix, the primary structure here, okay, the secondary structure here. So it all form tertiary. These are all in one unit forming tertiary structure. Okay, another stabilizing force for tertiary structure is disulfide, which don't forget that there are two amino acids that have sulfur, okay, in their side chain, so they can form disulfide bridge here, okay, if they happen to be closely to one another. Okay, this is the monomers. Okay, here is example of cysteine monomers. Okay, the last structure of protein is quaternary. So this is representing one unit of tertiary. So when you have more than one unit of tertiary close with one another like this, it will form quaternary structure. Okay, so the stabilizing force would include all the listed in the previous slides. Okay, so those are all the stabilizing force for quaternary structure. So example of protein having quaternary structure would be collagen, which are fibrous type of protein, okay, and then hemoglobin, which is globular type of protein. Okay, so this is the summary. We have the primary structure here. Okay, this is the alpha helix of secondary structure, and this is the beta pleated sheet of the secondary structure, and then both this secondary and primary structure will form tertiary structure, and more than one unit of tertiary structure, so more than one unit of tertiary structure will form quaternary structure. So that concludes the first part of protein.